So, essential question. It has been the same the entire chapter. What are living things made of? We went from elements to a very simple compound in water to bigger compounds, the macromolecules, the 4 that we talked about last week, and then enzymes. Enzymes are a specialized class of proteins that we focus on because they are very important to living things, especially us and inside of our bodies. So, our key concept today: enzymes are catalysts for chemical reactions in living things. We will define that word in a minute. And I'm not sure if I've used them before, but I like to use GIFs when things are a little bit harder to visualize in your brain. I'm a visual learner, so I like moving pictures. We are going to talk about all those parts in the pictures in a minute. Catalysts. All reactions need a certain amount of energy to activate or get started. This is the activation energy. You guys got that right in the chat earlier. Very good. Catalysts increase the rate or speed of chemical reactions by decreasing the amount of activation energy needed for a chemical reaction to get started. So we call them catalysts in a general sense, kind of in a chemistry lab or even in nature. There's lots of catalysts. And this is a typical graph. Good morning, Alexander. Where you have the amount of energy on the left, and you have substrate to products that represents some sort of chemical reaction. Something is turning into something else. And with no enzyme, you need a lot of energy to get that chemical reaction going. But with an enzyme in the red, you only need this much energy to get that chemical reaction going. So with an enzyme, that amount of energy needed is decreased. And that helps all of our chemical reactions in our bodies and elsewhere move a lot faster. That's okay, Alex. I hope your teeth are good. I hope you got them cleaned. Enzymes are specific catalysts that are found inside of living things. So when we talk about enzymes inside of our bodies and other living things, we call them enzymes. They're needed for almost all processes that happen in our bodies. Most of them are proteins. There are a couple exceptions, but in the general biology class, we talk about the general class of enzymes. And then there's examples of chemical reactions in our bodies that use enzymes. Good morning, Brianna. Digestion is the typical one. You eat your food, you chew it up. Your food becomes a different form as you chew. You have enzymes in your saliva that start to break down that food while it's still in your mouth before you even swallow it. And then you have more enzymes in your stomach, in your small intestine, in your large intestine. It breaks down different parts of your food as it processes through. Metabolism is when our bodies suck the nutrients and the energy out of our food and put it back into our cells. Those require different enzymes. Muscle contractions. Every time you move, the fact that you're sitting up straight, your spine, your muscles are helping you sit up straight. Those require certain enzymes. And then bone formation. Most of you are still growing. You have growth plates in your knees that are stretching out. And for them to stretch out, enzymes need to catalyze that chemical reaction to form more bone in your long bones and your legs for you to stretch out. These are all chemical reactions that require different enzymes. The lock and key model. So we can't see enzymes with our eyes. They're too small. So scientists have made models. An enzyme structure only allows certain things to bind to it. Each enzyme is like a lock, and there is only one specific key. The key, when we're talking about chemical reactions, is called a substrate. Those are the fancy science name for the key in a chemical reaction. The place where the enzyme and substrate come together and actually touch is called the active site. And you can see two different kinds in the picture. So here the substrate comes in one piece. It binds to the active site, and then it splits into two. That is a chemical reaction. And in the end, it's called products. The products float off and go do whatever it is they were meant to do. The opposite can also happen. So on the other side, you have two substrates. They come together in this active site, and then they bond and form one product. And then that one product that is now bonded together floats off and does whatever it's going to do. So there's two kinds of chemical reactions that happen in our bodies. We break stuff up or we put them together. Both of those happen with enzymes. Enzymes themselves do not change. So during a chemical reaction, as you saw in the last slide with the GIFs, it's not the enzyme that changed. It's the substrate that came in and either got broken apart or put together, not the enzyme. Enzymes are always floating free in our bodies to speed up as many chemical reactions as it can. And this is a very typical example of how enzymes and substrates come together, where they come together, how they end up. This will help you in your this picture will help you in your next assignment. It will help you on your quiz. It will help you on your test next week. You don't have to draw this if you don't want to. You know where it's found. But you have the enzyme. You're going to have to label that. You have the active site. This little part right here where the enzyme comes in. These are the substrates. These are the keys. This is the type where you start with two substrates. When it comes together, it is called the enzyme substrate complex. And then it is now bonded into a product. It releases, it floats off. You can see nothing at all happened to the enzyme. The enzyme stayed the exact same. It's going to float off and catalyze another chemical reaction to make as many products as it can. Disruptions. Enzymes function best within a small range of conditions. All enzymes have optimum conditions that they work best at. I don't know if you remember the story of Goldilocks and the Three Bears, or if you ever told that story, but Goldilocks is a little blonde girl running through the forest, comes upon a cabin, breaks into the cabin. She's hungry. She sees there's 3 bowls out, 3 bowls of porridge, which is kind of like oatmeal. She tries the first one. It's too hot. She tries the second one. It's too cold. She tries the third one. Anyone remember what the third one was? Anyone remember the story? Well, the third bowl of porridge was just right, so she ate it all up. Just right. Yes, Fisher. Good. Yep, that third bowl is just right. So she ate it all up. So that just right condition, there's more to the story, but you can look that up if you want to. That just right condition, not too hot, not too cold, that in science is called the optimum. It's our fancy science name. And conditions that affect enzyme activity. You have to have a just right temperature. You have to have a just right pH. We talked about the pH scale. Some enzymes actually like it acidic. That's their just right. Some like it basic. Most enzymes like it just about neutral. Our blood is neutral-ish. So most enzymes that work in our bodies work at whatever our pH of our blood is. And then ionic conditions. Salt is an ion. Enzymes like just a little bit amount of salt, just like we do on our food. We don't want too much salt because then it's too salty, but you have to have some salt or else it'll be bland. So we have to have a just right amount of salt to be able to function properly. All right. That is it for these notes. These notes are fairly short, and that is also it for the chapter. So make sure you finish these notes today. The recording will be on Canvas later today. You can still take them with the blanks. Be sure to include your summary. There are directions on how to do the summary on Canvas if you still need it, or if you're new. Work on the enzyme labeling assignment. It should be really short, and you should be using that picture I showed you. Just what part is the enzyme, what part are the substrates, where's the active site, what are the products, be able to pick those out of the picture. Let me know if it works for you or not. It should work for you. And then take the 2.5 check for understanding quiz. That is the last quiz. Later today, I'm going to post an extra credit lab. It's a virtual lab about enzymes. Some of the videos didn't come over, so I have to re-record the videos really quick before I post it. But I'll post that later today, and you can work on it this weekend if you want any extra credit. But it does have to be done before your exam next week if you want to do it. And then your Chapter 2 exam will be the first thing you do next week, and then we'll move on to Chapter 3.